All right, thank you so much. My name is Ben Forrid. Let me see if I can get this slide to advance here. All right. Well, big thanks to the organizers for inviting me. This is a really big honor to participate in this panel discussion. I also like to thank you for coming. I know there's a lot of great talks and stuff going on this afternoon, so I appreciate you coming to spend time with the panel today. And a big thanks to Global Genes for live streaming all of this stuff, giving folks that aren't able to join in person the opportunity to still participate and see what's going on. And there's a remote chance that my wife and kids are out there watching right now, so hi, guys. Today we're going to talk about registries and natural history studies. And I think as a panel, what we decided to do early on when we were talking about this is to set a foundational talk to give you guys kind of an idea, if you're just dipping your toes into the water here, of what types of things to consider when making decisions and kind of what it takes or what goes into building a registry. So we hope to demystify some of this stuff for everyone today and hopefully come away with some good information. If not, we are going to try to leave ample time at the end for a good discussion so that there's good back and forth Q&A. So please be thinking of questions as we're going through our presentations. So the first thing to start out with, what is a registry? In the most general terms, a registry is simply a collection of contact information and names for people with a given disease. And you do this in the hopes of being able to reach out to those folks again later on to make them aware of research or clinical trial opportunities. The data here is often provided by patients themselves, but there are times when clinicians or physicians can enter data as well to provide their input. And you're going to hear more about that from Ashley later on when she talks about natural history studies, which are different than registries. Registries are not simply an email list, something that you kind of put together and have in-house. When you talk about a registry in the technical sense, we're talking about a defined study, something that requires informed consent and is usually overseen by an institutional review board or an IRB. And the AHRQ it's an agency under the Department of Health and Human Services, much like the NIH or the FDA. The AHRQ put out a big old book on how to build a registry, the types of things you should consider when building a registry, and we kind of lovingly call that the Bible. When we're working with stuff, we consult it when looking at the elements to consider, and that book takes a lot of account into the ethics of building a registry. Someone who participates in a registry has to consent, and you have to record that consent and so on and so forth. So as you're looking at building this sort of things, keep that stuff in mind. The registry itself, however, can be as simple as a spreadsheet. So it doesn't need to be a big, fantastic thing with all these bells and whistles. It can be pretty straightforward and simple. But depending on what sort of information you're asking and what type of data you'd like to collect, it may require some more sophisticated software. Depending on the volume of data you're collecting, it might require server space. And so you can make it as complicated as you'd like. Why are registries important? The first big one is when your group is approached by a pharmaceutical company or by a researcher who has an opportunity for a clinical trial, their first question is going to be, where are the patients? Where can we reach out to folks and make them aware of this opportunity? And I think you want to be prepared for that when it happens. It also provides you a way to reach out to your community members. So from a foundational aspect or from the community the patient advocacy groups aspect, being able to collect data that you can use for marketing or for educational purposes to educate your community, also to raise awareness for your community and put out marketing materials and things in doctors' offices and so on is a use of a registry. From a researcher's perspective, I guess I didn't go into this too much, but my background is in cell and molecular biology. So I spent a good number of years working in a laboratory studying what's going on inside of a cell. And from a researcher's perspective, we look at things like cells growing in a dish. And it's this kind of abstract notion. We gain information from that. But an opportunity might come up for us to apply that information to a given disease state. And that doesn't make us an expert in the disease. In the rare disease community, the expert is you. The patient and the patient's family are the people who become the experts in the disease, and it's that story that should be driving the research. And so when researchers find a way to apply what they're learning through their basic investigations to a given disease, having a registry available to them so that they can go in and say, how do I steer this ship? What types of things go on in this condition? What are the things that I need to be aware of before I dive in to look at what's going on at a molecular level, is really big for a researcher. And then finally, when pharmaceutical companies or researchers are moving out of that translational stage and they're looking at opening up clinical trials, they need to define inclusion and exclusion criteria and all of that fun stuff. And having a registry there, a collection of data that can guide them in those decisions, is really helpful. So what's the best way to launch a registry? I think the simple answer to that is whatever is best for you. There's not a cookie cutter way that you can go into launching a registry. There's some things that might work really well for one group that won't work for another, and so on. On here, I've put together just a few different instances or a few different options that people will commonly use. The first one may seem like the most straightforward out of the gate, just to go ahead and do it on your own, but that can be more complicated than you might initially think. There are obstacles like finding everyone, building up the registry database itself. There's IT costs that go into that, and it can become quite a bear. And that's something that we've worked with groups on as well, as they've kind of started out on that path and then look to maybe work with a different partner. You can work with a researcher or a clinician team. So maybe in your community, you've got a rare disease researcher that focuses on your condition, and you're looking to provide patient samples that can then be correlated to a data set that lives in a registry. That becomes this really important and valuable tool for that researcher. If you have that relationship with someone, that might be a really good option for you. For other groups that might not have that physician champion or that researcher champion, maybe something a little more broad would be something that you'd consider. And then thirdly, you can farm this stuff out. You can work with groups that specialize in this sort of thing, and there are a multitude of them out there. I've listed some of the bigger ones here on the board. NORD, of course, has the I Am Rare registries. Genetic Alliance is the peer registries. Invitae has the Patient Insights Network, their genetic testing company. And then represented on the panel today, we've got Ashley from UPenn, the Orphan Disease Center registries, and I work at Sanford Research with the CORDS registry. So you can approach us and work with us to build up the registry infrastructure. This is something we've done and we've replicated time and time again across multiple different disease states. So the last thing that I kind of wanted to go through in my little spiel here is just a case study, something that you can use to use as a foundational basis. My only experience with registries is working for CORDS, so that's what I'm most familiar with. So we'll use this as an example, but other groups will be slightly different or similar to what we're doing, and I encourage everybody to kind of shop around, make sure that you find the best solution for your community. The way that CORDS operates is by offering what we call a standard questionnaire. And so anybody who goes to sanfordresearch.org, clicks Enroll Now, will be presented with the same data set right up front. And it's basic contact stuff. We ask some demographic and socio-demographic information. There's a brief section on disease, diagnosis, symptoms, where you were diagnosed. You can type in information there. And then we can ask really important quality of life questions too that are a little bit harder to quantify. And in the most basic sense, that data will go into the CORDS registry and will be freely accessible to researchers. The thing that we do kind of pride ourselves on, or being able to build upon that, is to work with advocacy groups to create disease-specific questionnaires where we're able to take it down to the next level. So you can imagine a person filling out the standard questionnaire and identifying themselves as having a rare condition for which we've partnered with an advocacy group. And they would be presented with that advocacy group's disease-specific questionnaire and really be able to drill down into some of the details that would be valuable for a researcher down the road. So you can ask questions on specific genetic testing results. Maybe there's multiple different mutations that cause the same condition. Maybe there are a specific trinucleotide repeat expansion that exists there that you can go into in that disease-specific questionnaire. Specific disease rating scales can be incredibly powerful tools for research. So I think of like the ataxia rating scales have 1 to 4 ratings or 1 to 5 ratings that you can mark down over time. And then we can ask specific quality of life questions, things that are harder to get out of a laboratory report. Instead of looking at enzyme levels, we can ask questions like how often do you have trouble sleeping at night, or how often do you feel sad? Those sorts of questions are just as important to give the researcher or give the clinician an impression of how that person's life is going. Now you can collect all of this data in one snapshot, and you get an impression of what that person's situation was at the time they sat down and filled out that questionnaire. What we like to encourage people to do is go in and fill that same questionnaire out every year, or as often as symptoms change. And in that way, you don't just get the one snapshot, you get a continuous story over the course of time. And when you multiply that across everyone who's ever completed that questionnaire, you can build a pretty good impression of what the disease's progression looks like. So that's kind of how we operate with CHORDS. And like I said, you kind of have to look around at what nuances work best for you and when evaluating your registry needs. So lastly, feel free to contact me. I'm an open book. I'm here to help you with anything that you need. I've got my email and phone number up here, so take a picture. All right, thank you.
Thank you so much, guys. Thank you, Ben. So I just wanted to say thank you for inviting me. I was really honored to be invited. When I know being here, there are so many patient groups and patient advocates that I really look up to. So at first, I wasn't really sure why they asked me to come and talk about patient registries, but Sanford has been a huge help to us, so I wanted to share that story with all of you. And sort of share our registry odyssey. I call it an odyssey because we've made a lot of mistakes. So you can I am by no means a registry expert, but I think my story is sort of the hot mess to success, you could say. Well, let's just say on the road to success. We're getting there. Anyways, so yeah, I was kind of laughing to myself when Global Genes asked me to speak, and thank you again for doing that, because I sort of hate registries as a patient advocate, as a foundation president. They are sort of, to me, the necessary evil of running a rare disease foundation. So. Okay, so I just wanted to give you a quick overview of our foundation. It's called the Pitt Hopkins Research Foundation. My son was diagnosed in 2012 with Pitt Hopkins. At the time of his diagnosis, there were about 200 kids diagnosed, and no research whatsoever. There was no other patient groups. There was nothing. Within a month, my family and I started the Pitt Hopkins Research Foundation. We raised about $400,000 that first year, and since then, we've raised over $3 million and funded research all over the world. We are the only foundation devoted to Pitt-Hopkins. So I know a lot of us advocates have multiple foundations in our group. You know, maybe there's a foundation that works with support, and maybe there's a foundation that does research, and maybe there's even another foundation that's handling the registry. We handle it all, which is why, at least at the outset, registry kept falling to the bottom of the totem pole of priorities that we needed. So I'm just going to give a shout out to, if we're live streaming, if any of my Pitt-Hopkins families watching, Pitt Strong, hashtag. We've grown a lot since then, and I know this is a chance at Global Genes for all of us to tell a little bit about our diseases and share with each other. So I just wanted to give Pitt-Hopkins a plug. It's a rare neurogenetic disease. It is not fatal, but it severely impacts quality of life. My son cannot walk, he cannot talk, he will be cared for his whole life. Imagine the symptoms of autism, epilepsy, cerebral palsy, breathing issues, all rolled into one little boy who can't talk, or one child who can't talk. So as daunting as this all is, I know you guys all understand why we keep plodding along. So in the first couple of years, we were super excited, and we got some researchers interested, and we're raising money, and they're like, well, we need some patient surveys. And we're like, no problem. And we are rallying, we're getting all these patient surveys done, and it's great, and we get it done at this one research group in the Netherlands, and then the next 6 months later, this research group in Texas wants one, and I'm like, well, the research group in the Netherlands just did this. Can't you use theirs? Oh, no. No, we could never do that. So I'm just thinking, that makes no sense. And then we start realizing we need a registry, because we can't keep having our population fill out surveys and then having these researchers ask the same questions over and over again. Even after year one, survey fatigue was already setting in. We had 3 big surveys done in the first year, and we were feeling really good about ourselves till we realized these guys aren't sharing any of the information. So we knew we needed a registry, but I'm super cheap, and I wasn't going to spend the money to do it. Also, time. I definitely felt like it was something that needed to be outsourced, because I don't know how to build a registry. And then hassle. Building a registry right, it's not the sexy part of rare disease foundation work. Not that there really is one, but you know, we all want to be like talking to the gene therapy guys and getting them on board and giving them money and on the phone with them. That's the most, well, not fun, but the stuff that keeps you going. The registry is like the workout you have to do to wear the bikini, right? So we created our own. So finally, I'm like, well, I'm not spending money on that. That's ridiculous. It's like people signing up for stuff. We can do this. Well, that was a big mistake. I don't even know what we were thinking, honestly. We didn't have IRB approval, but we had our own consent, which I don't even know what that meant. We didn't even have software built for it. It was like Ben was saying, it was like on an Excel spreadsheet. I mean, it was a little bit better than that, but it was, you know. And then as time went on, we realized we weren't asking all the right questions. And honestly, I don't think with a registry you ever ask all the right questions. There's always new questions that present themselves, and every researcher wants different questions. And we started realizing that, and that became a real issue that we realized we're gonna have to figure out how to tackle that. And then the way we set it up, you know, our vice president set up, she's amazing, but on the back end, we realized the information was becoming really difficult to mine. It was very difficult how we set it up. So we start thinking, we've got to outsource this. So we're looking, and then this one place is like, yeah, we can do this for you, but we're going to charge you a whole bunch of money. So they were out, because I won't pay for it. Then we won this grant, and they were going to build a registry for us, but yet they still wanted us to pay a little bit, and we were going to be the first of a handful of registries they built, and the more we were on the phone, it just didn't sound like the right fit, and we just didn't want to be the guinea pigs, and it didn't feel right. So Now, meanwhile, we have our registry on our site, and it's growing, and we're getting like we have like 300 people who've registered, which is pretty good for a rare disease of, at this point, like maybe 700 people. So we're feeling pretty good about our information, and we don't want to lose all that information that we've already gathered. So another year passes, and we're getting more patients, and we know our registry isn't great, but we don't know what to do, and we're doing more research, and now we're really getting ready for clinical trials. So now it's like 4 years later, we're really getting ready for clinical trials, and people are saying, well, what about your registry? And we're starting to realize these holes, the ones that I already mentioned. And a woman that I met at a conference like this was like, have you heard of Sanford? And I was like, no, but they're going to charge me, and no. She's like, no, they're totally free. And I'm like, no one's totally free. They're totally free. So we went with Sanford, and this is why it worked for us. They had done it before, and they knew what they were doing. So I mentioned that other one that was just kind of starting out, and I think it will be good, but we didn't want to be the guinea pigs because we had already messed up once. We needed to know that we were going to be in good shape. It was free. They provide guidance. They provide IT support. If one of our patients is logging on and they can't figure out something's not working, I don't have to fix it. I just email Austin, Sanford, you deal with it. It's easy to mine. We own the data. And most importantly, it's easy to make adjustments. So if we miss a question, we can go back, or if we see something start to trend in the data that emerges, we can go back and easily email all the participants and say, hey, can you go back in and add some more information? And that was really important to us too. So we launched our Sanford registry in June of this year at our conference, and it's doing well. I would say we already have about 50 participants. What we need to do now is really market it. So we're launching a marketing campaign this month, and we're going to do it on social media, on email, on Facebook. And then the thing is, once we do this marketing launch, it's just a constant, as you guys know, we have to constantly remind people to sign up. We did a good job with the first one, but the first one wasn't as long. And that's another thing that I really liked about Sanford. It's longer and it's more detailed, but it's not crazy long. One of the registries that we were working with wanted to have a base of 400 questions. And I was like, no one's ever going to fill that out. I won't fill that out. So the person that we worked with at Sanford said, look, you really can't go above 10 pages because no one's going to fill it out. So they really guided us, and I think in the end we came up with a really good registry that hits the most important things but isn't ridiculous long. So in addition to that, some other things that we're doing that I wanted to share with you guys, and they're here today. Maria Picone, is that Maria? Blair. They're here today, and I know Maria, who's the founder, is speaking, but we're really excited to get involved with this company called Trend Community. And they have a bunch of platforms to mine and aggregate patient data. But the one that I'm most excited about is they've figured out a way to get IRB approval to go into your Facebook groups and mine the data in your Facebook groups. And they have a special algorithm that searches keywords and key phrases and key symptoms, because that was the most frustrating thing. This is why I get so frustrated with registries, and when everyone's always telling me you need registries, I'm like, I can tell you everything you need to know. Just ask me. I'm right here. I can tell you. Give me an hour. You'll have a full picture. We don't need this. Of course, that's never good enough. But with Trend Community, all that information that's liquid gold in all of our Facebook groups, which I'm sure we all have, they're mining it. And we haven't fully gotten this all set up yet, but that's our next step. They're mining it, and so that researchers can also access that data. So yeah, I already just said all that, so there you go. I forgot I made that slide. And then there's another group called Backpack Health that we're looking into. Similar, the patients sign up, you input your data, And you, again, it's all IRB approved, and they have ways to share it with researchers, health professionals, all that kind of stuff. Truth be told, I need to look into this more, but I just wanted to share it with you today as something to maybe go home and research. So now we have a registry. Like I said, it's constant marketing. We send out emails, we send out newsletters. One thing we're gonna do that we did with our old registry, which was really fun, is a raffle. So we got a big screen TV around Super Bowl time. Everyone was really excited about this. We got it on Black Friday for really cheap. And we got a huge, big flat screen TV, and anyone who filled out the registry was entered to win. So that was a big boost. And we'll probably do that every year, honestly, just to get people to fill it out, refill it out. And you can do it multiple times a year too. You know, to buy a big screen TV is like $300 on Black Friday, so not a big expense, and it really paid dividends for us. So that's our natural history story. I hope you can learn a little bit from our mistakes and our successes and from these great people here today. Thank you so much.
All right, so I'm going to try to catch us up on time so there's ample discussion opportunity at the end. So one of the things I'm tasked with is talking about natural history studies, and that falls in this lovely area of, I know what a registry is, but why do I need a natural history study and what role does it play? So I'm going to try to divide that terminology up a little bit, but I would say there's admittedly a very gray area still. So one distinction I want to make is a natural history study really is, it can be a registry on steroids, but there's really a longitudinal component. That's key. If you take away the database part, a lot of these things can run on the same database or data collection structure. So let's take that out of the equation for a moment and just think of natural history studies as a way to understand disease symptoms over time. Longitudinal, it's really important. So I'm going to introduce you to the terminology we use at the Orphan Disease Center, because I know a lot of people have different jargon and different backgrounds that they're coming into this space. I'm formally talking about natural history studies at the bottom to mean the collection of clinical data under formal clinical protocols during a special research visit. Now, I'd say there's a gray area of informality with natural history studies where you operate in a research registry space is kind of our term for it. You're collecting data as a parent or a patient in a web portal or questionnaires, but you also want to integrate that clinician entered data. Anything that really gets at what the progression of symptoms are over time gives you a picture of natural history study. Some of the questions are, do you operate more formally or informally? So I'm going to talk a little bit more about the formal natural history study and what that means and why maybe at some point your community wants to work in that direction. So in that formal setting, a goal for a natural history study may be to develop a comparator data set for clinical trials. Hugely important for clinical trials where there might not be a placebo arm. And that's rare, and you'd say, oh, we don't have that, but gene therapy is becoming much more common. Gene therapies do not have a placebo arm, so how do you tell if if the therapeutic is having an effect on symptoms? You can compare it to natural history. The other aim or goal for your natural history study can be to inform clinical study design by identifying and testing clinical outcome measures. So that's important to evaluate new endpoints and understand patient or family burden. Potential to collect lead-in data. Now that is data that if you're going into a clinical trial, you can have what's called lead-in data. Maybe it's 6 months of that patient before they go on a therapeutic intervention. That's what we call lead-in data. So natural history can also serve a purpose of trying to bridge that gap before someone goes on an intervention. And these are a little bit of innovative trial designs that are becoming more popular, but I'd say they're a little bit in a different kind of regulatory conversation that maybe Francis can speak to. And then to characterize clinical changes over time with respect to regional differences in standard of care. We find that rare disease is treated very differently by different specialists in all regions of the world, and understanding that standard of care in Asia and how it differs from maybe the US can be really important when we go into those countries and try to run clinical trials. Understanding that background is really important for where a patient comes into that clinical trial. They may start at a very different starting point. So what does this look like in reality? So if we get away from the data capture mechanism, usually a formal natural history study, in our view, will mirror a clinical trial visit. So, for example, we have one study where we're running an in-clinic portion, that kind of formal clinical collection of outcome measures. And I'll go into that in a second, but we're also pairing that with a web portal. We think that's important because we can still collect patient-reported outcomes from a wider kind of swath of the population that maybe can't participate at the trial site. We also want to test the boundaries a little bit of this informal and formal data collection mechanism to see if we can get the FDA to kind of talk to us about that in-between gray area for rare disease. So stay tuned. That'll be interesting to see what happens. For one disease we're working on right now, it's a 2-year study that we're doing natural history on. We have 5 research clinic visits per patient. Each is 6 months in frequency. So you have a screening visit. In another 6 months, you come back. Another 12 months, 18 months, and 24 months until the end of the study. It's a full-day study visit. You have a full clinical protocol that the clinician and the site coordinator coordinate with you in advance and then walk you through a full day of measurements. And actually, for this one, we try to operate internationally wherever possible. So we have 3 sites in the US and 3 in Europe. And then to give you an idea of assessments, when you walk into these visits, we usually do a medical history, physical exam, quality of life assessment, which is really hugely important to pair quality of life or burden of disease for the family with your clinical outcomes. And then the clinical outcomes can be things like functional measures, how long does it take you to walk across the room? How fatigued do you get? Measurements of language or respiratory function, whatever is really very important for that disease. It may include imaging, MRIs, X-rays, ultrasound. And whenever you can pair this effort with biosampling and collect samples, plasma, urine, CSF, that is ideal. CSF is a hard sell to patients. We understand that. So anytime that is an opportunity to be collected, we really value that, but that can be a hard sell. And at least from our perspective, we are not an expert in any one of these diseases. We're really just trying to get data and get it back out to the community. So in order to build our natural history studies or registries, we work with the community every step of the way. So we incorporate patient groups, and that is both at individual level and within foundations through a patient-aligned structure, and that's really an effort to organize that community around registry and understanding endpoints that are relevant. We then work with industry, researchers, and clinicians in our kind of clinical working group and try to bring them together early at the table so that we know that the data is going to be used by industry. And we're very transparent with the community that when we collect your data, when you agree to participate in these studies, it's for use by the entire community. That's academic researchers and industry. That's hugely important. If that is something of concern, then maybe it's not the study for you. And then we work with, kind of build those groups into a scientific advisory board for each of our studies. We try to identify a coordinating site for the study, and then we build the database and data capture mechanism on top. Now, how do you get at data variables and endpoints? It's kind of an important part of this entire process, and you could spend an entire session on this alone. But you work with the community again, and that's starting with maybe a lit review, so looking at the literature and what's been published, and oftentimes for rare disease, there's a lot of gaps. So then it's going back to the community. Has there been a registry built? Is that a good starting point? Identifying from the community key questions, and that's talking to patients, parents, and clinicians along the way, saying, what are your concerns? What do you know? What do you not know? What are the gaps? And then it's, hold on. Then it's working with key stakeholders, again from that map before, in an iterative process to make sure you're not losing things along the way that are really vitally important to the group. And then it's building consensus across that community. If everyone's not bought in and convinced that this is relevant, then you're gonna lose half the group down the way. Either the patients aren't gonna participate, or industry isn't gonna see the data as useful, or clinicians aren't gonna see the value in their own participation and time. So really, you've gotta bring the whole group together along the way, along that journey. And then, importantly, if you can evolve, if you can work in a platform and with a group that has the ability to evolve over time, that's very important, because what you're gonna find is you start with a small group of rare disease patients that represent this part of the spectrum, and then you start to bring in more participation and find your spectrum is huge. You want to evolve your understanding of the disease over time. So when you try to look at what endpoints are relevant, you have a questionnaire of 500 different data variables as a starting point. That's probably not going to have great turnout from your community if you decide to collect that either in the registry or in the natural history study. Trying to capture all of that in a one-day visit is very hard. So where do you start? I think there's different ways to go about this, but I would say we kind of focus on a nesting doll model where we start with what is the most timely prioritization. So if there is a clinical trial on the horizon and industry is asking to understand what endpoints are relevant, I would start at the bottom. This becomes the focus, is your current trial endpoints. This allows industry or academic researchers that want to move into the space to move very quickly without being held up while the community tries to understand and really drive research in the background. So I would prioritize that, but if you're not there yet, you might want to start with your patient-reported outcomes, or maybe, I think patient-reported outcomes or really research questions, which aren't represented here, but I think kind of falls into a similar area. It all depends on really that internal debate of what are your priorities as a community, where are you at this point in time, do you have time to really step back and ask questions, or do you kind of need to push forward with clinical trials? So arguably, if you're in the bottom kind of middle part of the nested doll model, start with the clinical trial endpoints and then move into patient-reported outcomes. But when you compare these together, it's much more powerful data. And then after you get a handle on that data, you can really move into exploratory and bigger disease characterization questions. And then just a few take-home messages and things that we've learned working in this space. Community registries or natural history studies should support multiple investigational interventions. So we as an organization, we partner with any industry partners in that space. We're not just committed to one partner, but natural history studies can cost a lot of money. So oftentimes we partner with a group initially to get a study running, but the data will be available to other partners who might come into the space later. Excuse me. We collect patient-reported and clinician-reported outcomes together in the same environment on the same patients whenever possible. We work with a platform partner that can evolve over time and move from patient-reported outcomes to clinician-entered data into that formal natural history study as well. And then always, always, always be transparent about funding, your intentions, and the use of the data afterwards. Without those things, you're going to lose the trust of the community along the way. And then one thing that I think everybody gets very kind of concerned about, and very understandably, we're a philanthropically funded group, so we often think about sustainability. So consider different funding models from the outset. Look at government funding, grants, industry, foundations, patient groups. All of these have been contributors to our efforts in the past, and I think that makes for a mixed portfolio of who you could reach out to fund. There's also the opportunity to think through different financial models of a consortia kind of environment where you bring multiple, if there's multiple foundations in a space, multiple pharma in a space, kind of bring together and pool resources. And then really consider what are your primary goals and features of that data collection mechanism up front, and then scale appropriately and enable future growth when maybe funds are more available. So I think that's it. If you guys have any questions, that's my email. Feel free to reach out.
I think I may need this. Perfect. you mind? Okay, perfect. Thank you very much. Hi, thank you very much for coming and listening to this talk. And I'm Francis Kalush, and I work for the FDA, for the Center for Drug Evaluation and Research. I'm part of a PACE organization, that's Professional Affairs and Stakeholder Engagement Group. And microphone. Okay, sorry. 4 to 5 years ago, our center director, Dr. Janet Woodcock, decided that it's very important to bring the external stakeholder voice into the drug approval process. Usually the divisions will be working with the sponsors, but not having any type of interaction or listening to the patient and patient advocacy groups and the professional societies. So we're really a focal point for advocacy and really a two-way engagement on the drug development, review, and safety. And as part of our role, we have run a couple of workshops, public workshops, and the goal of those public workshops was to educate patient advocacy group, and even professionals, healthcare professionals, about the drug development process. And you could see that we had an annual Navigating CDER workshop, and I would encourage everybody here to go into the website, and all those meetings that were video recording, and also we have transcripts. And I think education is power, and more for the patients and for the patient advocacy groups. Because at the end of the game, what everybody would like to have? You would like to have a drug that will cure or treat the rare disease. And also we had a meeting on diabetes outcomes, and in collaboration with Global Gene, we had a meetings on rare diseases. And at that meeting, we discussed different case studies. And really, one of the main goals, or two take-home messages that I wanted to emphasize on that meeting, it was building registries and natural history studies, and also to show the flexibility of the FDA when approving drugs that we call it orphan drugs for the rare disease community. And it was really, the meeting demonstrated the beneficial overlap of effective advocacy techniques and FDA regulations. So this was the meeting that was held last year in collaboration with Global Genes, and it was attended online and in person for more than, I think, 600 people. So we also have new legislation. How many people know about the 21st Century Care Act? Very nice. So should I go over this information or should I run? But the most important is really that a report on the use of the patient experience data, and that's the reason that it's so important to build those natural history studies and data and the registry, because that information is gonna be really used on the patient experience data. It's gonna be used in regulatory decision making. And that's the opportunity for you as patient advocates to come and have your voice heard in the drug development process and approval. So let me give you a little bit about the new drug therapy approvals. And we have different mechanisms in place for the approval of drugs. And there are some that really show how can you use the different designations. And just to let you know that out of the new molecular entities, the novel drugs that CDER approved in the last 4 years, between 40 and 42%, there were orphan drugs for the rare disease community. So just you are educated on those numbers. This report is online, and there are different designations. Some, like 61% of all the drugs, they use any type of expedited review pathways. You have the accelerated approval, the priority review, shortening the time of the review from 10 months to 6 months. Also the breakthrough therapy, and also the fast track. And the fast track is really, there is a lot of communication between the FDA and the sponsor, and we are not waiting for the end, for the whole submission to come. FDA will review it as portions of that submission will come to the FDA. So a total of 61% of all the novel drugs that were approved using any of those designated expedited pathways. So, and now, the most important part for this community, it's not that we were on time with the goal date for the review, but if you look at the red bar, 39% of all the drugs approved last year in 2017, they were drugs for orphan diseases. And 78% of those, if you look at the blue bar, they were approved first in US before any other country. And I know that there are a lot of misunderstanding about how FDA approve and if we are really innovative on the way that we are thinking, but we are, and I'm gonna be showing you more data here. So that's the data for the last 4 years, that 41 to 42% of all the drugs that they were approved, the novel drugs, they were for orphan drugs. They will fall into the category of orphan drugs. So what is a rare disease? Do you guys know what's a rare disease? Because many of the different terms that people have been talking, it's ultra-rare disease, but defined by the FDA, we don't have an ultra-rare disease definition. We wish we would have, and maybe we would have more regulatory flexibility for approval of drugs, but there is none, and there's been a lot of going back and forth and meetings. But defined by FDA, really, the disease, for drugs and biologics, the disease needs to have a prevalence of less than 200,000 in US. And worldwide, it changes. In EU, it's less than 5 per 10,000, and in Japan, less than 50,000. And there are different examples of low prevalence rare diseases. Some of those are more known: cystic fibrosis, Duchenne muscular dystrophy, and ALS. But I'm going to be talking about the flexibility that the FDA used to approve 2 drugs in 2015 for hereditary orotic aciduria, that it's a very rare disease. You may consider it an ultra-rare disease, and also for hypophosphatasia in my next slide. But before I show you the table, so there are different flexible approaches, and those can include, you were talking about 2 adequate studies, right? But really, based on the regulation, we have the flexibility that the approval could be supported by fewer than 2 adequate and well-controlled studies, and there are a lot of examples of that. We are using the accelerated approval pathway, as I showed you before, for all the novel drugs. There are new novel trial endpoints. And hopefully, the rare disease community is going to be bringing those PROs, the patient-reported outcomes also, as novel trial endpoints. And then your voice and your experience living with the disease is going to be part of the drug development process and the clinical trials. And also the use of non-concurrent controls. And that could be used using natural history studies information, even publications. And really, CDER reviews are really open to talk with the community, so you can contact us. So this is a table. I try to simplify it. So there are the 2 diseases, hereditary orotic aciduria and the hypophosphatasia. And they have enzyme defects. And there are patients reported in medical literature. For HOA, there were 20 since 1959, and 15 well documented. And for the HPP, there was a natural history clinical study data. And there were 2 drugs approved for each of the different diseases in 2015. And they were using all the different designations that I mentioned earlier. They were designated as orphan drugs. They were using the breakthrough therapy, the priority review, the fast track, the breakthrough therapy, really to shorten the time for the review. And so we will have a drug to treat those 2 diseases. But the most important part, and showing the flexibility I want to show you, that's for the new drug application data. For HOA, it was based on 4 patients and published literature information. And there were a lot of discussions with the clinical reviewers at FDA and the applicant. And for the hypophosphatasia, there were 2 prospective single-arm trial, and they were open label. They weren't even blind. Okay, so that shows the flexibility and how the FDA really would like to help. And based on regulations, because we cannot act without basing the regulations on our discussions and in the drug approval. So this is a slide that shows that the overall survival for Strensiq versus historical control patients. So we use historical control patients to compare it to the drug for the approval of Strensiq. Oh, sorry. So what are the final thoughts really here? To hear the patient voice and stakeholder voice, and we are open. And we have mechanisms, different tools that you can use to contact us. And you will have my email address also at the end. And patients' experience really can inform the drug development in many ways. Understanding the disease burden from the patient perspective, helping identify specific symptoms or disease manifestations that are relevant to the patients. Not that they're relevant to the clinicians, but relevant to the patients or to the caregivers. And really helping and maybe bringing new endpoints in those clinical trials that they were going to be of benefit to the patient or the caregiver. And really help to identify the benefits that may not be too obvious, really, outside observers or readily measurable. So this is my email, and thank you very much for giving me the opportunity to bring the message.
Okay, so we're going to move into the question and answer portion. I'm going to start with one from the app, and then raise your hands if you have questions. I'll bring the mic to you. So the first question that I have on here is, how do you help families who may not have health literacy to complete registries by themselves?
Well, I think that we, at Sanford anyway, with CORDS, when there's an existing questionnaire that's out there and folks are having difficulty navigating it from a medical literacy standpoint, they can contact us right from the form. There's a phone number that they can call, and we're available to answer questions and guide them through that process in any way that we can. It becomes a tough thing when you have these disease-specific questionnaires that, in some cases, ask very, very specific medical term questions, and providing the right answer is just as crucial as any other question. And so, being able to guide the individual through the enrollment process is really important. I don't know if do you have any?
Similarly, we try to write our registry questions in a way that is more accessible to a lower kind of reading level than I would say. I think there was mentioned in the last session during data that most consent forms are written on a graduate level, which is not really accessible. So I think the recommendation is on a 3rd to 5th grade reading level. I think there's ways to assess that. You can throw things into, I think it's on Google. I'd have to look up the tool. If you're interested, I can dig it out. But the other thing is really important is when we build our questions, we send it back out to the community to get their input. We ask that not just maybe a foundation head that we're working with very closely, but we ask them to identify people in their community that maybe not as well versed as the head of a foundation. So we try to access different people in the community to help us build, to try to keep that in mind. And then translation wherever possible is really key.
Yeah, I would echo that and just say we really did spend a lot of time. Like, we don't use phrases on our questionnaires like apnea, even though that is very common in our kids, and you would think a lot of our parents would know it. But we use breath holding, and we describe how you write the question. We found in our first go was so important, and you have to keep it really simple. And then we do have a lot of times parentheses explaining what we mean if we say, does your child have seizures? And then we explain this means blah, 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 blah. And then we describe the difference between absent seizures and tonic seizures. And we put the parentheses and explain everything, because you're right, yeah, that is really hard. And if there is a question that we don't think our families will answer accurately, we cut it.
I had a question. Audrey, I have a question for you. How do you tackle the challenge of being able to, or does your registry allow you to translate the questions into different languages? Because, of course, as you all know, we all have international families that can participate in our registries, and that's one of the biggest challenges we have. So, of course, right now we're in talks trying to figure out how to tackle that challenge to at least get maybe 3 of the most common spoken languages that we have in our patient group. But then that's a challenge in itself. So right now we only, the IRB requires us, they have to be able to read English. And what that's done is like, it's cut down some of the patients that have access or can understand the questions. Or it's not that they're not required, but they need to be able to understand English is one of the clauses in there. So had you had that?
More question for Ben.
Oh, okay.
We haven't run into that much, honestly, yet. But I do believe Sanford offers translation services.
So being able to communicate with people in other languages is across the board issue, because IRB, like you mentioned, will have regulations on how you can reach out to them. And if you translate the entire platform, IRBs require that it's forward and backwards translated. So that, from my understanding, that means I translate that into Spanish, and then you have someone who's fluent in Spanish translate it back into English, and you compare the 2 English versions. There's some legwork that goes into that to verify that the people who are reading it in another language are getting the exact same information that they would be getting if they were getting it in English. And so the way that we work around it is we use what we call short forms, which are basically, I think we have a dozen or so different languages that are in there, and presents a brief statement in the native language that explains what CORDS is, what the registry is asking them to do, what they're consenting to, the big pieces, and then it asks them to find an English-speaking translator to go through the whole enrollment process with them. And right now, through our IRB, that's kind of the workaround that we have. But I think that, like you mentioned, as the world becomes smaller and as technology becomes much more involved, groups like ours are going to try to face that translation down. How can we translate our whole platform to provide it to as many people as we can?
Okay, so I think we have time for one more question.
How do you handle privacy issues? Do you de-identify the patients? And I have another question. And do the patients have access to the information on the registries?
Sure. So the first question for our perspective and for Ashley's is we definitely de-identify everything that goes out. When things come in, there are 18 elements of identifiability that you have to clear, and it's name, address, age, all of this other stuff. Actually, age isn't one of them, but you de-identify the whole data set so that there's no way that you can trace it back to any one individual. So that's something that we take that very seriously. Do the patients themselves have access to the data in the registry? They don't get like a report or anything that per se. In our registry, they can go back and see all of their answers that they've done in the past. Like I mentioned, we asked them to go in and update it as often as their symptoms change, or at least once a year. And so they can see every iteration of the questionnaire that they've filled out beforehand. And then we also have this kind of handy tool that we're developing right now that allows them to compare their answers to other folks who have answered the questionnaires. Ashley.
Yeah, privacy is huge. Definitely, you have to be able to keep things private, or you're going to lose trust of the community. So we de-identify any data. A big part of what we're trying to do is get data back out to the community, so we'll de-identify before we share data. We limit those who have eyes on identifiable information to just the study team for the purposes of collecting data. And I think actually one of the problems with the new GDPR European regulations is actually need to have data be transportable or portable. So we're working on what that means exactly for people to be able to take away their data. But one of the important features we want to give back to the community for their participation is visualizations in our patient portal. So similar feature, trying to understand where you fall on a spectrum of kind of the aggregated data set for that community.
Benjamin, Francis, Ashley, and Audrey for providing us with your knowledge and expertise. So at this time, before you head out to your next session, we would appreciate if you could jump on the mobile app and take the survey about this session to provide your feedback. Our next breakout session will begin at 3:15. Please use the next, I think you guys have like 10 minutes, actually, 12 minutes to transition to your next session. Check out the map and agenda, and please ask any of the volunteers if you need help getting to your next session. Thank you.